So today we're going to be doing the rest of lecture 8. I wasn't able to finish in my morning class. I expect, we're going to end up at a similar spot here for the afternoon, but that's okay. I mean, like, there's no quiz next week. We have plenty of time to catch up. So the first thing we're going to talk about today is Mendel's second law. You don't have to pull up the PowerPoint. I'm gonna explain to you what's going on. We'll be writing a lot of notes today, and his second law. We talk about his first law, right? His first law was called the law of independent assortment, and now we are going to. Sorry, his first law was the law of segregation, which means if you have two chromosomes, because we're diploid, you only can pass on one of the chromosomes, right, to the next generation. And now he has a second law, which is called the law of independent assortment. Okay, so let me just explain what that means to you first. I mean, you can read the definition here, but it's much better if I show you an example, right? So law of independent assortment. Okay, which basically means this. If we take a look at genes on a chromosome. Let's say we have two homologous chromosomes right here, and that's big A and small a because they happen to be different alleles. And here we have big B and small b. So according to his first law, right, the law of segregation, you have big A and small a, but you can only pass on either the big A or the small a, not both, right? So that's the first part of the law. Right? You have big B and small b. You can only pass on big B or small b, not both. That's the law of segregation. Now, the law of independent assortment says that whether you pass on the big A or small a will have no effect on whether you pass on the big B or the small b. The best way to think about this is imagine a coin. Okay, so I have a coin, you have a coin. I flip my coin and then I get heads. Whether I get heads or tail is not going to affect what you are going to get on your coin, right? My coin is independent from your coin. The outcome of my coin is going to be different, independent, not different, but like independent from the outcomes of your coin. Okay, one has nothing to do with the other. That's basically what is happening here. The outcome of gene A, as in whether you pass on the big A or small a, has no effect on whether you pass on the big B or the small b. Okay, think of the A as one coin and the B as the other coin. All right. So this person here is big A, big B, small b. Let's think about what kind of gametes we can create. Right? So the gamete is going to have an A and it's going to have a B. Right? Not going to have the same letter because you only can pass on one of the A's and one of the B's, right? The only time you're going to have two of the same letter is if the chromosomes fail to separate in meiosis. We talked about the last class. It's called non-disjunction, right? So we don't want that. Now, what kind of gametes do we have? Well, we can have big A and big B. That's one possibility. That's one gamete, right? This could be like, a sperm or something like that, right? Big A and big B. What's another combination? Help me out here. What's another possibility besides big A, big B?
Big B, B, big B, big A.
Sorry, one more time. Which one is it?
Big B, big
Big.
A.
Yeah, but that's the same thing, right? Big B, big A is same as big A, big B, right? Isn't it? Right. The order doesn't matter, right? Okay, so here's another one. big A, small b. Okay, big A, small b. Right? And then we can do small a, big B. And then as Matri said, small a, small b is also possible. Okay, so in this particular case, there could be 4 different gametes. So let's try another one. This is example 1. Example 2. Let's say the person is big A, big B, small b. What are the possible gametes? What are the possible gametes? That's correct, Blessing. We have big A, big B. What's another possible gamete? That's right. Big A, small b. Right? Now we can try the other one. Look, big A, big B, but we already have it. So we don't have to draw it again. Big A, small b, we already have that as well. So we don't have to write it. So for this particular case, there are only 2 possible gametes. Gametes are sperm and egg, right? In the case of flower, it will be like, sorry, in the case of plants, it would be pollen and egg. Okay, let's do one more. What are the possible gametes? Is that all? Anything else? Okay. Big A, small b. Big A, small b. Big A, small b. Big A, small b. That's all. It's all just big A, small b. I only have to write it once. Okay. That's all. That's figuring out the gametes, right? Whether you get the big, like, just because you inherited the big, sorry, just because you inherited the big A does not necessarily mean you have to inherit the big B as well. They are independent. They are 2 different things. One is not going to affect the other. That's basically the essence of the law of independent assortment. Okay, so the next thing we're going to do today is dihybrid cross. Dihybrid cross. That took like a whole hour to explain. So if you are lost along the way, just stop me and tell me what you want me to clarify. Last class we did monohybrid, which is a single trait. We're only tracking one thing. In dihybrid crosses, we are tracking 2 things at the same time. Okay, tracking 2 characters. 2 characters. Character is like, flower color, seed shape, seed color, so on and so forth, right? So I am just gonna copy this thing over. The 2 traits we will be looking at is seed shape and seed color. Okay, seed shape and seed color. Round is dominant over recessive. Sorry, round is dominant over wrinkle, and yellow is dominant over green. Okay, round is dominant over wrinkle, yellow is dominant over green. And what we're trying to do here is, or what Mendel did was, he crossed a pure round yellow and a pure wrinkle green. Two things we're looking at, right? So this is the parent generation. They are pure for all the traits. So we have couple names for that. And we can call them purebred. We can call them true breeding. Right? All these means the same thing. Homozygous, it just means that they are the same allele. Right? That's what pure means. Okay. So we have two traits. We have the seed. What do we call it in the PowerPoint? Is it type or shape? Shape. Okay. Seed shape. And we have seed color. Dominant, recessive. So round is dominant, right? So we use big R and small r for wrinkled. Yellow is dominant and green is recessive. So that's what we're going to have here. All right. What is the genotype of pure round yellow? Can someone please tell me? What are we going to write for the genotype, the genetic makeup of round yellow? Well, round is big R, right? And they are pure. So that's big R, big R. What about yellow? What's the genotype for the yellow part? Are we confused? Yes, big Y, big Y. That's right. Okay. Because it's pure, they have to be same. And, it's round and yellow. That's all. For wrinkle green, that's little r, and then little y, little y. Okay, so let's take a look at the gametes. The gametes of the parent generation, the P generation. Right, what kind of gametes are we going to get from this parent? We just practiced earlier today, right? So, what are the gametes from this individual, from this plant?
Big house. Big.
Big R and what?
And small house.
No, we're just looking at this person. There is no small r here.
Okay, big R and big Y.
That's it. Big R and big Y. That's all, right? And then it's the same for all the other ones. Big R, big Y, big R, big Y. That's why I only have to write it once. I'm just going to put a bar here to separate it. What about this parent? What are
Small
the
house,
possible? What's
small
that?
house, small house, small Y.
That's right. Small r, small y. Okay. Those are the gametes. So if we do a small table to figure out the different combinations, right, we are going to put the big R, big Y here. And we're going to put the small r, small y here. And we can see 100% of the offspring will be big R, big Y, small y. Which means, what do they look like? Are they round or wrinkled? Well, because they have the big R, right? They are going to be round. That's right. And the color, they have a big Y. They will be yellow. So round and yellow. So I want to point out something here. I'm just going to do like a side note here for you. Like a side note. I will write it in red so you know it's important. If both parents are true breeding, okay, like this case, right? They're true breed, round, yellow, wrinkle, green. All F1 generation individuals will have the dominant phenotype. Okay. See, both of these plants are pure. They are homozygous for both R and Y. But when you breed them together, the offspring, right? The offspring here, they look like the dominant trait. Because the wrinkle is being covered up, and so is the green. So I'm gonna give you an example here. Okay, let's say there is two kind of mouse here. Okay, so this is mouse number one. Mouse number one is going to be let's say it's gray. Okay, gray. Okay. And then it has a short tail. Gray and short tail. And then we're gonna have another mouse. Okay. This mouse is white and long tail. Okay. Let's say they are both purebred. So they're gonna make some baby mice. And the baby mouse look like this. So my question to you is, and this is an important question, which one is the dominant trait? Is gray dominant or is white dominant?
Gray.
Gray is dominant. That's
Yes.
right, because it shows up as the F1, right? Whatever the F1 shows, that's the dominant trait, right? So gray is dominant. And what about tail? Is the actually, you know what? Let me make this like super long. There, that's what I want. Is the long tail dominant or is the short tail dominant?
Long tail.
Long tail is dominant. Okay, so the phenotype of the F1 will tell you which trait is dominant in the organism. Does anybody have any question about this? You need to know this, otherwise you will waste a lot of time on the quiz to figure out what's dominant and what's recessive in some cases. All right. So let's go back to our main note here. We are done with the F1 generation. Okay, so F1. F1 is R, R, Y, Y, all of them. Okay, so what we're gonna do next is we will take two F1 individuals and breed them. Right? So that's one individual, and then that's a second individual. And they are the same genetically because 100% of the F1 looks like this, right? So now I want you to take a moment and write the gametes. Okay? Write the gametes for each of the parent. So the gametes, that's right. A lot of you have been typing it in the chat box. So big R, big Y, big R, small y, small r, big Y, and small r, small y. I'm going to put a line here to separate it from the other parent, but it's the same thing for the other parent. Right? So our table is going to be 4 by 4. Right? So take a moment and make a table, if you could. Let me see if I can make this bigger. Just give me a second, okay? There we go. Okay, so that is my table here. And I'm gonna put the gametes on the top and on the side, right? So I just copy it. Big R, big Y. Okay, you just copy them, right? The gametes. I'll give you a moment to set up this table. Please send me a message when you're done. So I can proceed. Just say ready. And then I'll go on. You don't have to fill it in, okay? We're gonna fill it in together. I just need you to draw the chart. Okay, thank you. So, two of you are ready. I assume the rest of you are ready or close to ready. So what we're gonna do is we're gonna combine these gametes, right? Like what if this gamete fertilizes this gamete? What are we gonna get? Well, it's quite simple. Big R, big R, big Y, right? And then we're gonna go ahead and do the next one. Big R, big R, big Y, small y. And we will just continue to do it. Okay? If you want, you can do it yourself and compare with what I have. If you are not sure how I'm getting the answers, you can look. So for the second row, right? It's a big R with a big R. So like this. And then it's a big Y with a small y. Right? So we just keep going. Matching up the letters and writing out the combination. Each of these represent a possible outcome for the offspring, for the F2 generation. Now, I made a mistake this morning and someone caught it. I'm not saying if I'm making a mistake now, but double check what you have with what I'm writing. And if there's something that looks a little bit off, please let me know. There we go. Okay, so there are 16 possible combinations for the F2 generations. Are we okay with the table?
Sir,
Mm-hmm.
doesn't matter the order you write it in. Do you always start with R?
Yes, I mean, like I just try to match it up with the same order, right? Otherwise, it gets a little bit confusing. Yeah, but like, and if it's like this, then we gonna put the capital first. Is that what you mean?
Yeah.
Mhm. Okay, so
So,
sorry,
you start with the rows on top first or the column first?
sorry, one more time
Like for the row, right?
for the row. Yeah, for a
You
row.
start, do you start with the row first when you're writing it, or we can start with the column?
You talking about the first column and the first row?
Yeah,
Yeah, it doesn't matter in this case.
doesn't matter.
Yeah, I mean, like I can show you like these ones. It goes right here, right?
Yeah.
Yeah, and then these ones will go right here. They just represent gametes from each parent, right? Is that what you're asking me?
No,
No?
no.
Okay, what do you come
Like
again?
for example, okay, let's go R,
Hmm.
small r and big Y,
Small R, big Y,
and big
okay.
Y
Yeah.
times
Hmm.
big R and big Y.
Yeah.
So can I not put um
Oh, I see what you mean. So like you're
small
saying
r
it
then a
like
big
this?
Y? Yeah.
Yeah. Yeah, I
Does
mean
it
it's
matter?
not wrong, but like generally people put the capital first. Yeah.
First?
Yeah.
Okay.
But I mean it's like I'm not gonna be seeing your work, so whatever makes sense to you. You're still gonna arrive at the same conclusion, okay?
Okay.
All right. So I am not gonna be doing the genotype, I'm just gonna be doing the phenotype. Okay, so we can have, let's look at the dominant. Big R. Big R is a round, right? You guys remember? And what about big Y? What's big Y? Big Y is yellow, right? Okay. So I want you to take out a highlighter or pencil crayon, whatever. Find all the round and the yellow in this table. So that's a round and yellow, right? Round and yellow. Okay. Go find all of them and tell me how many boxes are round and yellow.
Eight boxes?
8 in total
Uh,
or 8 more?
Well, I just did it based on the capital letter.
Yeah, that's a good strategy. Anyone else? Did other people also get 8 or you guys have other numbers? Okay. Well, this one is round and yellow, right? This one is round and yellow. Anything that has a one big R and one big Y, that's going to be round and yellow. Right? This one too. And this one as well. That's all. Okay, so if you count all these boxes, it is going to be 9 out of 16. 9 out of 16. Okay, so maybe you are missing a box or you have an extra box. Just make sure that you got them all. Now, it's not surprising that there are a lot of things that are round and yellow because round is dominant and yellow is also dominant. That's why we have a lot of them, right? Let's try another one. Let's do round and green. Okay, so round is dominant, green is recessive. How many of these things are round and green? Okay, please find out and let me know. That's good, Entry. So round and green, that's this one. Green is little y, right? Round and green. And round and green again. Okay, so that is 3 out of 16. The next one is wrinkled and yellow. Okay. Wrinkle is recessive, yellow is dominant. Wrinkled and yellow. How many are wrinkled and yellow? Okay. Just 2? Anyone else? 3? That's right. All these are wrinkle and yellow. Wrinkle and yellow. And finally, the most rare form is wrinkle and green, which shouldn't be surprising because both of these traits are recessive. The only time when they show up is when the dominant is not there, right? So that's really rare. 1 out of 16. Okay. So the ratio is 9 to 3 to 3 to 1. This is an important ratio to remember. Okay. The most common form is dominant. The middle 2 is for dominant recessive dominant. And then the most rare form, the rarest form, is recessive. Okay? That's all. Anybody want to ask me anything? Before we do another question? Okay. This is from the PowerPoint, all right? I'll read it to you. In bees, normal wings are dominant to vestigial wings. You don't even have to know what it means, okay? All you need to get from that sentence is, okay, wings, normal or not normal, okay? Normal or vestigial, okay? But if you want to know, vestigial means like tiny wings. And then deep pollen sacs are dominant to shallow pollen sacs. Okay, so that's the first sentence. And we're going to stop there. Okay, and then we're going to write down some information. Remember, we are doing dihybrid cross questions, which means we have 2 characters to track. In the previous example, we were tracking the round versus wrinkle and then yellow versus green, right? So in this new example, what are the 2 characters we are tracking? Well, according to what we just read, we have wings and we have sacs. And we got to figure out the dominant and the recessive. The question spells it out for you, right? Normal wings are dominant to vestigial wings. So big N for normal, small n for vestigial. I stress the point that you don't necessarily have to know what it means to do the questions. Okay, sometimes you have these weird words in the question that talks about a trait. You know, no need to look it up. You just write it down. Right? Normal, not normal. That's what it goes on in my head. What about sacs? What letter should we use for the pollen sacs? For the dominant? Any suggestions?
Big S.
Big S? Yeah, sure. Okay. Anyone else? No, I heard someone say something else. No? Yeah, we could use big S. We can look at for me though, I would use big D. Okay, because then like it says deep is dominant over shallow, right? So I don't want to use I know why you chose S though. You chose S because it was talking about the sacs, right? So like, you know, first letter, right? But then there's also like shallow, the word shallow. So just for me personally, I don't want to use S to confuse myself, right? So I'm just gonna use big D for deep, but it's really up to you. You can use, you know, A, B, C, X, Y, Z. It doesn't matter. As long as you can keep track, it's okay. Okay. I would advise against using things like O, because like sometimes people write the O and then sometimes they're rushing and then they cannot tell whether they're writing big O or small O. And then it gets confusing. I also have people who chose L like on the test, like big L, small L, like that's just asking for trouble, okay? So choose letters that are easily distinguishable. All right, that's good. This is the table. You should have one of these for every questions so you don't get confused. Now let's find out who the parents are. The proud B parents. We got the male B here, okay? And then we have the female B. Let's deal with the male first. Let's deal with the male. Here's the male B. What's going on with the male B? The male bee has vestigial wings and shallow pollen sacs. Well, vestigial is recessive, so the question doesn't need to tell us whether it is homozygous or heterozygous. If it's recessive, it must be. It must be homozygous. Little n, little n. What about the pollen sacs? It's shallow, right? Shallow. And shallow is recessive, so I know it's little d, little d. Please write out the genotype for the female bee, homozygous normal wings, and heterozygous deep sacs. Please give me the genotype when you have it. Thank you. That is absolutely right. Is homozygous normal, so big N, but for the deep pollen sac, blessing, because it says heterozygous. Heterozygous means it must be different, right? The two alleles. So that's why it's big D, small d. If they say homozygous, big D, homozygous deep sacs, then it would be big D, okay? So that's it. All right, we got our things all lined up now. We're gonna write the gametes. The gametes. Okay, so for the male bee, it's just little n, little d, right? No other choices. What about the female bee? What kind of gametes? Right? Big N, big D, big N, small d. Yeah, that's all. Right? So our table is going to be pretty small this time. Our table is not like 16 squares anymore, right? This is just 1 by 2. Right? Something like this. Right? So we're gonna put the male one here. It really doesn't matter where you put them as long as you can keep track. And then this is big N, big D, big N, small d, right? So give you a moment to fill this in, and we'll take it up in, I don't know, like a minute or so. Okay, thank you, Tahib. So let's take it up. That was a minute. So big N, big D, small d. Big N, small n, and then small d, small d. So in terms of the genotype, we're gonna have 50%, 1 out of 2 boxes, right? 50% like this, and 50% like this. And for the phenotype, 50%, what kind of wings are they gonna have? Right. See, sometimes it's hard to keep track. That's why we roll it down in the table, in the chart, whatever you want to call it. We roll it down at the beginning, okay? So what is this one? Is it normal wing or vestigial wings?
Normal.
Normal wings, that's right. Normal wings. And then deep or shallow?
Deep.
Deep. Thank you, Sarah. Deep sacs, right? And then the other 50% also normal wings. And shallow. All right. So the table is not always 4x4. That's the worst case if you want to think about it, right? That's like the biggest one, and it's so much work. But sometimes it's just 1 to 2 or 2 to 3 or whatever the case happens to be. Okay. Anything you want to ask before we do the next example? Alrighty. I hope this makes sense to you. This is the next one. All these questions are in the PowerPoint, okay? I'm just putting it over here so that I can post the solution online after. Assuming curly hair, big C, is dominant to straight hair, small c. Okay? So we know we're talking about hair. Dominant, recessive. The question tells us curly is dominant and small c is straight. Okay? I know there are other kinds of hair, but within this question, that's what it says. If we keep reading, it tells us albinism is a condition in which cells which normally produces pigment does not do so. A person with this condition is called an albino. The allele for skin albinism, small n, is recessive to the normal allele n. So the other character we're looking at is skin. We have big N for normal skin and small n for albinism. Okay, so people with albinism, they don't have pigments. So they are very pale. Their eyes are blue. Hair might be blonde. Hair is blonde, actually. It's like even if you are like of Eastern descent, right? Asian or Orientals. If the person has albinism, they would again have like very pale skin, blue eyes, blonde hair, right? So that is the situation here, and we have a woman, and then the man, and then some kids. So usually it's good to represent these kind of complicated situation with a family tree. In genetics, family tree is called pedigree. Pedigree. All right. So square is male, circle is female. Right. This means to have kids together. So let's try to represent this situation with a pedigree. Okay, we have the woman with the man, and then they have a child. We don't know if the kid is boy or girl. Let's just say they have a girl. Doesn't really matter. It's not going to change the question. The only time where it matters whether it's a boy or girl is if the condition is inherited on X or Y, which is what we're going to look at later. Okay, let's look at the woman here. What's going on with the woman? Curly hair and albinism. So curly hair is big C, but I cannot tell right away whether she's big C, or big C, small c, right? And albinism, little n, little n. For the guy, straight hair and normal skin pigment. So normal skin pigment is big N something. We don't know if he's big N, or big N, small n. But we do know he has straight hair, right? So straight hair is like little c, little c. Let's take a look at their daughter. The child has straight hair and is albino. Some people say albino, actually. So what is this? Well, if she has straight hair, is small c, albinism, small n, small n. Okay. So she has two small c. One of the small c must have came from dad, right? Well, the other small c must have came from mom. That's how I know the woman is big C, small c, and not big C, big C. Similarly, if you look at the skin allele, one of the small n's has to come from the mother. And then the other one, of course, has to come from dad. And that's how we know the father is big N, small n. Okay. All right, let me copy the other question over. How many unique gametes could be produced through independent assortment by individuals with genotypes of that? So this, we are looking at 5 characters, right? 5 genes at the same time. For dihybrid, we're only tracking 2, but now we have 5 different genes. Okay, so how many different alleles, how many different combinations can we generate? So for example, we can have big A, big B, big C, big D, big E. That's one gamete. I can have big A, small b, big C, big D, big E. That's another gamete. Okay, so the question is asking, don't write these down. Okay, the question is asking you, how many different combinations can you get? What do you guys think? How many different combinations?
Two.
Sorry, how many? 32, did you say?
Yes.
Okay. Anyone else? Give me some numbers. No? All right, that's fine. So let's see how we do this. Well, let's take a look at the gene A here. We have 2 choices. We can pass on the big A or the small a. 2 choices. What about gene B? Also 2 choices, right? Big B or small b. What about C? Well, C is homozygous. We only have big C's. That's the only thing we can pass on. So that's just one option. Big D, small d, 2 options. Big E, one option. So if we multiply all these together, 8 possibilities, 8 gametes. Okay. What about now? What about this? How many different combinations can we get? Okay, 10. Remember, we multiply, not add, okay? Okay. Very good. So 2 options here, 2 options, 2 options, 2 options, 2 options. Multiply all of them together, 32. All right. Next one. Consider the cross, this and this. What is the probability of creating this? Okay. So this is like a tri-hybrid cross, right? Like 3 gene at a time. Can you tell me how many gametes from this parent? You don't have to write them out, but like just tell me how many gametes for this parent. You should be able to tell me after the previous 8, right? Exactly. So there are 8 gametes for the first one and 8 gametes for the second one. That means if we're gonna do that table, the Punnett square, it's gonna be 8 by 8, right? 8 by 8 is like 64 combinations. That's like way too much. Okay. So what do? Well, let me give you a quick lesson on probability. Okay, so side note again. Side note. Probability is very easy, okay? Don't worry. Let's say I have a dice and I have a coin. Okay. How many outcomes for rolling a dice? 6, right?
6.
Yep. And how many outcomes for flipping a coin?
2.
2. That's a tail. Okay. So I want the probability. Probability means the chance, right? Probability of rolling a 5 and getting a tail. Rolling a 5 and getting a tail. So what you want to do is this. You want to figure out what's the chance of getting a 5. What's the chance of getting a 5? In fraction. What's the chance of getting a 5 when you roll a dice?
1.
Sorry, how many?
0. 1.
Oh, yeah. But in a fraction, 1 out of 6, right? Right? 1 out of 6. Because there are 6 possible outcomes, right? Only one of them is a 5. Yeah. What about getting a tail? I should say and here. Okay. Getting a tail is 1 out of 2, right? 1 out of 2. So I want to know what's the chance of getting a 5 and a tail happening together. We just multiply the probabilities together. Okay. So this works if the events are independent of each other. Okay? Meaning the outcome of the dice has no effect on the outcome of the coin, right? As long as the 2 events are not related to each other, you can just take the probability of one and multiply by the probability of the other to figure out how likely is it for both of them to be happening at the same time. Okay? So back to this question, we have 3 genes, A, B, C. According to Mendel's law of independent assortment, the first thing I taught you today, the outcome of A is independent of the outcome of B, which is also independent of the outcome of C, right? So it's like the dice and the coin, they have nothing to do with each other. Which means over here, if I want to figure out the chance of getting this genotype, I can just do the gene separately. Right? So let's focus on A first. A is big A, small a, big A, small a. So that's easy enough. If I have a table, I'm gonna make this thinner. It's not fitting. If I have a table, I can put big A, small a, and a big A, small a, and we can put them together. Right? This should be straightforward to you by now, right? So what's the chance? What's the probability of getting big A, big A?
1/2.
1 over what, sorry?
2.
2? I want big A though. Right?
1/4.
1 over 4, that's right. It's right here, right? 1 over 4. Does that make sense, Tahib?
Yes.
Okay. Now I can do the same chart for B, right? I'm not, I mean, you could do it later, but I hope you understand it's the same chart. But I switch all the A's to B's, right? So the chance of getting big B is also 1 over 4, right? And then the chance of getting big C is also 1 over 4. Right? So what's the chance of all these happening at the same time? That's what the question is asking, right? All these happening at the same time. It will be 1 over 4 times 1 over 4 times 1 over 4, which is 1 over 64. Okay, so it's much easier to do it this way rather than doing like a big table, 8 by 8, 64 outcomes. It's hard to keep track, right? Okay. So this is the next question. We're gonna do it after the break. I mean, if you don't need a break, feel free to try it during the break. But I think it's good to, just walk away from this for a little bit. I'll leave this on the screen, and then we'll come back in 5 minutes, and then I'll take it up. Okay? See you in a bit. All right. Let's do this question. Anybody have an answer they want to share though? If you do, feel free to type it in the chat box. But something like this is really it's possible on the test, okay, and on the quiz. So here is how you are supposed to do it. So here we have 2 true breeding stock of pea plants. True breeding. That means they are pure, right? One parent has red and axial flower, and then the other one has white and terminal flowers. Right? So here we have color, and we have type. Right? Again, I stress the importance of you don't need to know what it means to do the question. I mean, it might help to visualize it, but it doesn't matter. All you need to know is something is called axial, something is called terminal, right? So dominant and recessive. I have no idea which one is dominant, which one is recessive, because the question didn't tell us, right? So we gotta keep reading. All F1 individuals have red and axial flowers. Remember the mouse thing that I showed you? Where is that? Right? Look. Whatever shows up in the F1, that's gonna be the dominant. Okay? As long as the parents are purebred, this is true. Okay? So over here, let's come back to our question. Which one is dominant? Is red dominant or white dominant? F1s are red. Red is dominant. White is recessive. What about the flower type? Well, F1s are axial. So axial and terminal. So that's the first thing you need to figure out. Now write all this down. And then it says 1,000 F2 offspring resulted from crossing of 2 F1 individuals. Because the parents are purebred, we know for the F1 is 100% big R, big A, small a, right? So now they're saying if we take 2 F1s and cross them, what's going to happen? Well, hopefully you remember, in this case, we're going to have 4 different gametes. So if we do the table is going to be 4 by 4, which means we're going to get 16 possibilities, right? Like the ones we did in the first half of the lesson. But you don't have to construct the table. All you have to remember is the 9-3-3-1 ratio. 9 is the most common, 9 out of 16. That's for the dominant and dominant trait. This one is for dominant recessive or recessive dominant. And this one is for recessive recessive, right? It's the rarest. So what do they want in the question? They are asking how many of them would you expect to have red and terminal flowers? Red and terminal is dominant recessive. So what is the fraction that we're going to use? Is it 9 out of 16, 3 out of 16, or 1 out of 16? We're looking for the dominant and the recessive combination. Right? Dominant recessive, it's right here. 3 out of 16. Okay. So 3 out of 16 out of 1,000. We're going to key that into the calculator, which is 187.5. Right? So we choose the closest number. It's 190. Okay. It's not exact, right? Because the sample size is not that big. Like if you flip a coin 100 times, you're not going to get exactly 50 heads and 50 tails, right? Like you might, but it's unlikely, right? So that's why the number is not exact. Do you understand? You don't have to do the chart. You just have to know like what kind of trade do you want. What if they want like red and axial? Red and axial? Which fraction am I going to use this time? Yeah, 9 out of 16, right? 9 out of 16 times 1,000. Okay. If they want white and terminal, then it's 1 out of 16 times 1,000. All right. Okay, you might want to revisit this again. Some of the questions are like that on the quiz. All right, moving on. So far, we only talk about one type of relationship between genes, right? Dominant, recessive. Dominant will cover up the recessive. So you know this by now. Big R is red. Big R, small r is also red because the big R is dominant, right? But it turns out there are other kinds of interactions between genes. One of them is called incomplete. Incomplete dominance. Yeah, I'll give you a definition and then I'll explain. Incomplete dominance is the situation where the heterozygous genotype has a phenotype. The phenotype is the physical appearance, right? Yeah, so just copy that down first. I'll explain to you what it means. All right, so let's say we have 3 different flowers. Big R is gonna be red. Okay, big R is gonna be red. Where's my red? Right there. Big R is red. And then small r is white. Right? Nothing is new so far. So these are the homozygous phenotype. Okay, homozygous phenotype. And then it says the heterozygous genotype, which is big R, small r, has a phenotype, has an appearance that's an intermediate of the other two. So it is not red and is not white. It's somewhere in between. So pink. Okay. This kind of interaction is called incomplete dominance. The question will tell you. Now there is another type, which is called codominance. Codominance. Okay, so in codominance, we have got the flowers again. And let's use the same. You know what? Actually, let's use it different. Let's say big B. That's blue flower. Small b is yellow flower. Right? So for codominance, the heterozygous phenotype, sorry, genotype, has both alleles being expressed. Okay. So it's not somewhere in between, like in complete dominance, right? If this was in complete dominance, you would get like a green flower. But no, this is codominance. So you would have both blue and yellow at the same time. Okay. The flower is both. Okay. So you will have like blue and yellow showing up at the same time. Okay. Maybe like, you know, yellow flowers with blue dots, vice versa. So this is big B, small b, yellow and blue. All right. So this is the picture in the PowerPoint. Let's see. Red and white. If it's incomplete dominance, it's pink. If it's co-dominance, it's a combination of both. I am going to give you an example and you're going to tell me which one is it. The father is very tall, the mother is short, and then the child is medium height. What is that? Is that incomplete dominance or co-dominance?
Co-dominance.
Co-dominance? Okay. Why is that? Maybe I
Um,
draw it out, maybe it's easier. Okay, like very tall, you know, this is an exaggeration, okay? But then this is the woman, this is the man, and then you have a kid that grows up to be like this. Okay, the child. Child. Is this
because it,
Mhm.
I don't know, from my understanding, from what I understand, it's like half of the mom and half of the dad, right?
It's
So
somewhere in between though, right? Isn't it?
yeah,
Yeah. If it's somewhere in between, then it's incomplete dominance, right? That's what intermediate means, right?
Mm.
Like it's not red and it's not white, it's pink. It's somewhere in between, right?
okay.
So if it's tall, it's short, and somewhere in between, that would be incomplete dominance. Does that make sense? Have I convinced you?
Yes.
Okay. I'll give you another example, okay, for the whole class. Someone has type A blood, and then the other person has type B blood. They have a kid, and the kid has type AB blood. Is this incomplete dominance or co-dominance?
Co-dominant.
That's right, because it's both A and B, right? Not somewhere in between, right? I don't even know what somewhere in between would look like. You know what I mean? Co-dominance. Okay, that's good. Let's do another question here. This is example 6 from the PowerPoint. There are some kind of flowers here called a snapdragon. Red flower is incompletely dominant over white. And it tells you in the question. Right, so you can write it out. You can go like big R is red. Small r is white. Normally, big R, small r would also be red. But here it says the heterozygous condition is pink. What will be the result if you take two pink flowers and cross them? So pink flower cross a pink flower. We can go ahead and do the table, the Punnett square, that's what it's called. Big R, big R, small r. So just do them like we normally would. So we have 25% big R, which is red. 25, 50%, sorry, 50% big R, small r, which is pink. And 25% small r, which is white. Okay. Let's do one more. a red bull crossed with a white cow produces a roan calf. A roan calf has intermingled white and red hair. Okay, so it's both white and red, but it appears to be, this thing called roan. It's like, there's no such thing as gray hair, right? It's just an intermingled black and white hair, right? It appears to be gray. So we can do this again. Go big R. Big R is red. Bull is just like male cow, right? Yeah, male, and cow is just female cow, I suppose. And the calf is the baby cow. Small r is the white one. And the one that is heterozygous, it's called roan. So if the roan bull is crossed with a white cow, what percent of them will be roan? So we do the table again. Big R, small r on one side. Small r on the top. What percent is roan? 50%. Okay. So in terms of the work and the way you solve the problem is virtually identical to monohybrid. It's how you interpret the answer that is different, right? Any questions at all before I move on to the last thing here? I just have one more thing to teach you before we go.
I don't really get it, to be honest.
Okay, yeah, sure. So what's the question? Like, which part do you not get, I should say?
I don't know. Maybe I just need to review the whole.
Is it the chart that you don't get, or like how to get
No,
the
how
no?
to get the chart.
Sorry, the chart?
Yeah.
Okay. Do you understand where these come from?
Um. No, not
Okay,
really.
sure. So let's move even one step further back. How about these ones? Do you know these ones?
Yes, because the dominant one, when you said it's
Mm-hmm.
big R,
Mm-hmm.
heterozygous means one's dominant and one's not. So that's a
Yeah.
big and a small R.
Mm-hmm.
Am I saying it right?
Yeah, yeah, yeah. Mm-hmm.
Right.
Yeah.
And the white is the not non-dominant. So
That's
those
right.
are small R's.
Yeah. So everything is good. However, in this example, there is no more dominant or recessive relationship, right? Because if it's dominant and recessive, this one should be red in color, right? Do you agree?
Yes.
Okay, but it's not red, it's pink. And how do we know that? Because the question says it is incompletely dominant, right? So that gives it away that we're no longer talking about one gene covering up the other, right? We are looking at a different kinds of interactions where when the big R and the small r comes together, it's no longer one covering up the other, but they blend together to create a pink.
Okay.
Okay. So in this case, is asking what will be the result of a cross between two pink plant. So according to our legend here, pink is big R, small r, right?
Yes.
That's why we have a pink here crossing another pink. It's not any other combinations because the other one is red and white, right? Up to this point, are we together?
Yeah, yes, we are.
All right. So we have the big R and we have the small r. But when the plant makes the pollen, it can only pass on the big R or the small r, not both, right?
Right.
That's why when we put it on the table, we have big R here and we have small r here. Same thing for the other parent, either big R or small r. So the question becomes, what happens if this big R joins this big R? Like if this pollen and this egg comes together, what's the outcome? Well, the outcome is big R, right?
Mm-hmm.
And so on and so forth. Is the table okay now?
Yes.
Okay, so we do the table the same way as we would as from before, right? But the interpretation of the table is different now, right? We look at big R, that's still 25%, and the color is red, right? Because we said big R is red, right?
Yeah.
The biggest difference is when we look at the big R, small r, before, like what we learned last time, big R, small r would have been red, right? Agree?
Yes.
But not anymore in this question because the question says it is
Pink.
incomplete. Exactly, it's going to be pink. That's why we have the pink here, right? And then
Okay.
we have it.
I
So
get
that's
it.
the only difference, right? Like I said, the steps are the same. The numbers are kind of the same too, but then the way you interpret it is a little bit different. All right. Okay, so I hope that helps. And
Thank
this
you.
is very similar. Okay, you just go through it, and if you lost, just send me email. Okay. All right. So I know that is already a lot of information, but there's just one last thing I want to go through with you. It's called sex-linked. Sex-linked conditions. Okay. These are carried on X or Y chromosomes. Okay. If it's carried on the X chromosome, both males and females can have it. If it's on the Y chromosome, only males can have it. Okay. So an example of an X-linked recessive condition is color blindness. Color blindness. Okay. So some people cannot tell. The most common type of color blindness is red-green color blindness. They can't distinguish the two shades, right? So that's carried on the X chromosome. But it's recessive, which means you have to have two mutated X chromosomes in order for it to show up. If you're female, okay? If you're male, since you only have one X, if you inherit the colorblind X, then you're going to be colorblind. So let me show you a few examples here. If I put a circle, is this a male or female? Have you guys been paying attention? Male or female for the circle?
Female?
Yep. And this female is XX. Yeah, both of the X chromosomes are normal. So she is a female with normal vision. Female with normal vision. Okay, we can shade it in. We shade it in. This female is XCXC. The C is the colorblind gene. So this person, because she has both colorblind gene, this is a female with color blindness. Okay, she's colorblind. basically. What about XXC? Do you think she's colorblind or she's normal vision? Normal vision, that's correct, right? Because the normal X covers up the disease X, right? This is a recessive condition. So this is, we draw it like this, okay? Half shaded in. So this is a female with normal vision. But she is a carrier. A carrier is someone who has the disease allele, but then they don't have the disease. Okay. I mean, it doesn't have to be disease. It could be like, you know, like just some kind of feature, right? Okay, a carrier. So when it comes to X-linked recessive conditions, female has a bit of a protection because they have two X's, right? So as long as they have one normal X, it's able to protect them. All right, let's do a guy. This would be a male with normal vision, right? No colorblind X. He's gonna have normal vision. Okay. For this guy, oh, what happened? Oh boy. For this guy, he's gonna be XCY. Right? Male are a little bit unfortunate in this regard. We only have one X. So if we inherit the colorblind X, we're gonna be colorblind. We don't have another X to cover it up. So this is a male with color blindness. Can male be carriers then? Can a male be carrier?
Yes.
How so? Remember,
Mm.
carrier means I carry the disease allele but don't have the condition.
Because you have the chromosome.
I
The
do. Yeah, go on.
because you have the chromosome that in you, the X and the C.
Yeah, I have the XC, right? But I only have one X, right? So if I have an XC, I am gonna be colorblind, right? It's not like the female where she can have the XC and still not have colorblind, right?
Right.
Yeah, so a carrier is someone who has one of the allele, the disease one, the colorblind one, but they don't actually have the disease. That's why we call them carrier. They carry the disease X, but they don't have it themselves.
Well,
Okay.
that was the question, right? If
Yeah,
the
so the
male
question is, can male be carrier?
carriers.
Yeah.
So
And
did
the,
I answered it?
so the answer is no. Okay,
No?
male cannot be carrier because for males, for guys, you either have it or you don't have it. You either colorblind or not colorblind. It's not possible for a male to have XC and not be colorblind. Do you agree with that? Because we only have one X, right? So if I have the XC, I automatically have color blindness. It's not like the female. If they have XC, they might be okay because the other X is normal, right?
Okay.
Yeah, men don't have an other X to cover it up. So men cannot be carrier.
So they either have it or they don't have it.
Exactly.
Okay.
Okay. For color blindness, I'm talking about. All right. Anyone else have a follow-up question? All right. I know it's a lot to take in. I will review this with you guys again on when do I see you guys again? Tuesday? Yeah, Tuesday. All right. But I'll do one more question first. Consider color blindness. Find the ratio for F1 generation. All right. So we have a carrier female and a normal male. What is a carrier female? Carrier female is XXC, right? One of the X's is colorblind, right? But they are not colorblind. The female is not colorblind. They don't have any vision problem because they have one normal X to cover up the other one, right? Okay, what about male? What is the normal male? Well, normal male is simple. It's just XY. So we're gonna do the Punnett square. Gonna do the Punnett square. Okay. So for the woman, they could either pass on the normal X or they could pass on the colorblind X. The C is for colorblind, right? What about the men? They could pass on the normal X or the Y. So if this sperm fertilizes this egg here, then we're gonna have a daughter. They're gonna have a daughter, XX, right? XXC, XY, XCY. So for these questions, we want to separate it into male and female when we interpret the results. Okay, so these are the boys, and these are the girls. Okay. So let's look at the daughters. Daughters. Are the daughters gonna be colorblind? Any of them gonna be colorblind, or like what's the interpretation?
No.
Everybody's normal vision? Is that what you're saying?
For the girls?
Yeah, for the girls.
Yes.
Yeah, you're right. Okay, so all have normal vision. Okay, but 50% carrier, right? Carrier still have normal vision, mind you. Okay, but they're carrier, meaning like it's possible for their kids to have color blindness. Okay, depending on who they marry, right? What about sons? What about sons? Are all the sons gonna be okay? Are they gonna be colorblind?
50% normal.
Yep,
50%.
absolutely. 50% of the sons will be normal vision. That's like the top, this one right here, right? Like the XY right here. And then the other 50%, colorblind. Okay. How about one more? One more. Last one, I promise. Okay. Normal male with colorblind female. Okay. Normal male is XY. Colorblind female is XCXC. Okay, XCXC. So for the men, they can pass on the X or Y. It doesn't matter whether you put the men on the side or on the top, okay? Like it doesn't matter. Like where the parents go will not change your answer. For the female, it is XC and XC, right? So XXC, XXC. I guess like if you switch it, then you shade them differently, right? Afterwards. I mean, but still, it's not going to change your answer. All right. So daughters, are the daughters going to be okay?
Yes.
Yep, 100% normal vision. but all carriers, right? All carriers. Sons? What's going on?
Colorblind.
That's right, they're all colorblind. 100% colorblind. Right? That's why I try to tell you earlier. If we are looking at an X-linked recessive condition, the guys are more likely to get it than the females. Right? Because female has 2 X's, and if it's recessive, the other X can protect them, right? Males don't have that additional X, so if we get it, we get it, right? You know what I mean? Now I'm gonna say this last thing, which might confuse you, but I'm gonna say it anyways. However, if we talk about X-linked dominant, X-linked dominant condition, okay, not recessive anymore, X-linked dominant, then females are more likely to get it than men, right? Because female gets 2 X's. If any of those X's is the disease one, they will have it. Okay, men, same. If we get the X, we're gonna have it, but then we only inherit 1, right? So relatively speaking, men would have a lower chance for, X-linked dominant. Okay.
So which one is the X-linked dominant?
Oh, like an example of a condition like that, or like,
Yes.
oh, I don't have an example. Sorry, yeah, that's a great question. I only know the the recessive ones. I will look them up and I will I'll tell you. You know what? Let's Google it right now. I should know this as well. Dominant X-linked diseases. Okay, I just Google it. And example. They have a bunch here, but I'm not gonna lie to you. I have never heard of any of these. Okay, so you can Google it. There's something called Rett syndrome. There's something called double cortex syndrome. I have no idea what they are. Okay, but they exist. Okay, they exist. All right, that's it for today. Now, there are no quiz next week. Next week we'll finish up lecture 8, and then we're gonna start on lecture 9. The quiz is gonna be in next Tuesday. It depends how far we go on the biotechnology. The biotechnology is the hardest lesson of the whole course. All right, so I don't know if we're gonna be able to finish that. Depends how much questions you guys have. I'm flexible in moving the quiz. Okay, I'll see what happens. This quiz, because it's so hard, like all these problem solving and stuff like that, I am gonna let you do 2 attempts, okay, for this quiz. You can do it 2 times, and only the best score is going to be recorded. All right, so after you do the first time, you can see your mark right away. If you don't like your mark, you can do it again. And if you do better, you get the better mark. If you do worse, that's okay. You get to keep whatever you got in the first time. So hopefully that will, give you a little bit of like a wiggle room, right? Okay. All right, that's it. Thank you, okay, for coming to class every week. I'll see you next time. Enjoy the rest of your week.
Have a good weekend.
Thank you. Bye-bye.